Good morning, afternoon, evening, whenever you're watching this. Could be worse things. So, anyhow, I'm going to try to get through this in one take. Um, I might have to split this up, but this is the last one of Unit 5. We're gaining on it, or the line is gaining on you. Either way, here we go. So, we're looking at deviations from Mendel's model. So, that's stuff like sex-linked traits and linkage. Basically, it's those two things mostly. So, what this says is that if they don't follow the ratios predicted by simple segregation and independent assortment, you can identify that by quantitative analysis. That is like heart and soul of what they're likely to ask you to do if they ask you about this topic. Okay, so you have the idea, right, early 1900s, that a chromosome has got about 1,000 genes on it, roughly, a human chromosome. We call them linked, right? And that means they're not going to be independent of each other. They're going to be inherited together. So results of crosses with linked genes are not going to show like Mendelian, necessarily not going to show Mendelian ratios in offspring like you would expect if they're not linked. All right, that was his independent assortment idea. Realize this, first of all, any kind of discussion of linked genes has to be talking about two traits, right? Because you can't just talk about one trait and describe whether it's linked or not. It's got to be linked to another trait, two genes on the same chromosome. So, okay, um, we can also use some of those numbers to calculate map distance. Now they threw that in there, I think, just generally for you to be able to say you can calculate map distance. And one basic idea, the further apart they are, the more they'll cross over and the more these numbers, and I'm going to do a practice question here. So just pointing out all the stuff that's in the blue boxes to you right now. Okay, so here we have, again, if there's two genes, and these are like a B and a BG gene, right? A B gene. What do you call a bee that's hard to understand? A mumblebee. Oh, all right, back here. Concentrate, back here. So here we have the idea that if crossing over happens, we can make all these different gametes that if crossing over didn't happen, we wouldn't be able to make. Look, because if these two genes are stuck together, then you should only be able to make a gamete with those two genes still stuck together. And if the other homologous chromosome had those two stuck together, you should be able to make gametes with those two stuck together. But these two show a recombination of the genes from when they started, right? And therefore could produce some variety in offspring that you wouldn't have without crossing over happening. Okay? So if we now use this as an example, there's those bees again. Okay, so if this individual with these genes had them on different chromosomes and they assorted independently, you would expect them to be able to make these gametes, and quite importantly, in equal numbers. There'd be an equal likelihood that the big A ends up with the big B as it ends up with the little b. Make all four of these gametes equal numbers. This parent can only make one kind of gamete, right? So you would expect equal numbers of individuals that had both dominant traits, the dominant A trait and the recessive B trait, the recessive A trait and the dominant B trait, right? And both traits recessive, right? But if these genes were linked and the A and the B, big A and big B, were linked on one chromosome, and little a and little b were linked on the other chromosome in this parent, right? You'd expect this parent to only be able to make gametes with that big A and big B linked together, or gametes with little a and little b. So you'd expect only these kind of kids, kids that show both dominant traits or kids that show both recessive, and you'd expect 50-50, right? But the reality of it that led him to this idea of crossing over was that you still get some of these kind of kids that have one trait dominant, the other recessive, right? And that has to be because these crossed over. Now, the gene map idea, or the chromosome map idea, is how many of these kids do you get? That relates to how often it crosses over. So if these kids, which could only have resulted in the crossing over of genes to produce these kind of recombinant gametes, if these kids in total number 10% of all the kids, right, then that means these genes look like they cross over 10% of the time. And that 10 then can be converted to a number like, okay, we'll say that A and B genes are 10 apart. 10 what? We call them map units or centimorgans. But here's the idea. If these genes are farther apart, I'll now put them farther apart, right? There's more chance that they can cross over if they're further apart, because crossing over involves them touching and switching somewhere in between. And if there's more space in between, there'll be more crossover. Like, let's say, 40%. 40%, right? That means that show they would be 40 apart. So higher numbers of recombinants mean genes further apart. I think that's all they would ask you. They would not ask you to do this math. So here we go. All right. So now we have sex-linked traits, right? And the pattern of inheritance of sex-linked traits can often be predicted from data, including data in a pedigree. They tell you that they might ask you, here's a pedigree. Does this trait look like it's dominant, recessive? And now they're going to tell you, does it look like sex-linked, or again, the opposite of that is autosomal? Because those sex chromosomes, the X and the Y, and specifically the X here, right? All the other chromosomes are called autosomes, right? So now here we talk about illustrative examples, and here's where they threaten you with this. The standard story for mammals like humans, right, and flies like those fruit flies are XX females and XY males, right? So that means, unlike Mendel's idea, right, if the genes on the X chromosome are not the same as the one on the Y, then males only have one gene for each trait that is controlled by genes on the X chromosome, right? They don't have a pair that separates, da 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 da. Okay, but here's what they threaten you with, okay? They tell you that, I might tell you a story of birds. And like birds have this situation, males have two of the same kind of chromosomes. We call them Z's, not X's, but doesn't matter what you call them. It's the same idea. Females are the ones that have two different chromosomes. So it's just a different situation, but if we explain that to you, then we should be able to expect you to work out a Punnett square kind of problem and answer questions about that for birds or people, depending on if you understand the concepts. So here we have the identified by quantitative analysis. So here was the classic fruit fly cross done at Columbia, where you had this wide-eyed male, right? They correctly figured the wide-eyed, the eye color gene must be on the X chromosome, and he's only got one of them, right? Whereas mom's got two of them, and her red eyes was dominant, and this particular mom had homozygous dominant, two dominants. So all of mom's egg cells have one X chromosome, in this case with the red-eyed gene, and dad makes sperm cells with the X chromosome and sperm cells with the Y. Only the ones on the X have an eye color gene, right? All of their kids then are heterozygous if they're females, right? They got two different genes, two X chromosomes. But if they're males, the one and only eye color gene they got was the red one that males always get from mom for sex-linked traits. So now if we cross these two, we got a mom now that can make two different kinds of gametes, and a dad that can always make two different kinds of gametes, but only half of them have an eye color gene. So in our normal kind of Mendelian cross, where our parental generation, right, was, I'm going to use A's here, was one parent was homozygous dominant, the other homozygous recessive. All the kids were heterozygous. Oops. Right? You would expect 3 dominant kids to every 1 recessive. That's the idea that told Mendel pairs, segregation. That's why I get 3 to 1. Well, here you also get 3 dominant red eyes to every 1 recessive. But if they were also telling you the story and told you the gender of these, it's the fact that only males were wide-eyed, which would be the quantitative analysis that would tell you, oh, this must be sex-linked. Because the analysis is always going to be about some kind of gender difference. Whereas you figure with any other autosomal trait, it's half the females and half the males will have these certain traits. It shouldn't make any difference whether they're males or females. Here it makes all the difference in the world. Okay, so um, here we have the same thing. So look now, focus. Okay, here we have a mom who's got the dominant trait and she's homozygous dominant, right? And if dad was, they told you he had the recessive trait and this was autosomal, this would be dad, right? He'd have 2 genes, right? And you would expect all their kids to have the dominant trait. Well, in this case, all their kids do have the dominant trait. So in this case, the fact that it's sex-linked or autosomal makes no difference in the results. And that's also true of this. If you were told we had a heterozygous mom, oops, sorry, make that an A, and a recessive phenotype dad, you would expect half the kids to have the dominant trait and half the kids to have the recessive trait. And that's what we have here, right? Dominant trait, recessive, recessive. However, if you're told information about their gender, you can tell that this must be sex-linked. But now if we look at this one, right? If we look at this one, we have the heterozygous mom, right? And the dominant dad. So if we had heterozygous mom and dominant dad, and the dad was homozygous dominant here, kind of like he is here, is one and the only kind of genes he has are the dominant genes, then you would predict that this would be all the kids would be dominant because they'd all get a dominant A from dad. But here we have some recessive kids, and we only have one of them, and it's male. They're all male. So anyway, in the pattern of who has what and how many has what, but more importantly, what gender they are, you can tell, you can predict that this is a sex-linked trait. Okay, so this is what I said about the fact that this could be a story where maybe the males have the 2 sex chromosomes that are the same and the females don't, right? So here's the idea about these sex-linked traits, whether you have this ZW system or the XY system, right? The gender, which has 2 similar homologous sex chromosomes, females if you're talking about humans, right? ZZ males if you're talking about roosters. The individuals that have 1 dominant trait and one dominant allele and 1 recessive will be called carriers. They won't show the trait, but they can pass it on. And more importantly, all right, possible question-wise, the individuals that have just 1 sex chromosome, therefore 1 of the kind of genes you're talking about, they are far more likely to inherit sex-linked recessive disorders if they are rare, which they usually are, like hemophilia in humans. But if we're talking about birds, it would be the females that would be more likely to show sex-linked recessive disorders.